Today, I will be talking about microRNAs, small RNA molecules that play a major role in controlling gene expression. Although they do not produce proteins themselves, they help determine which proteins are synthesized and when they are synthesized. Here we will explore how microRNAs are produced, how they regulate target genes, and how their activity is controlled within the cell. We will also look at why they are important in normal biology and human disease. And finally, highlight some of the key research gaps and future directions in this rapidly evolving field. So now let's begin right at the start and look at how microRNAs are produced inside the cell. First, microRNAs are produced as longer RNA transcripts called primary microRNAs, and these are synthesized in the nucleus just like messenger RNAs. Next, a complex called Rorschach cleaves these transcripts by cutting them into shorter hairpin structures called precursor microRNAs. This is the first major processing step that prepares the microRNA for further maturation. After that, these precursor microRNAs are transported out of the nucleus into the cytoplasm. This export step is controlled by a transport protein called exportin 5, which carries them through the nuclear pore. And finally, once precursor microRNAs reach the cytoplasm, another enzyme called dicer processes them by trimming them into short, mature microRNA strands. At this stage, they become functional and are ready to regulate gene expression. Now that we know how microRNAs are produced, let's see what they do in the cell. Once the microRNA is mature, it does not act alone in the cell. Instead, it gets loaded into a protein complex called RNA-induced silencing complex, which acts as its functional partner and helps it regulate gene expression. The RNA-induced silencing complex guides the microRNA to its target messenger RNAs. This happens through sequence matching, where the microRNA recognizes complementary sequences on the mRNA and binds to its target. When it finds a target, it can either block translation or completely degrade the mRNA. Either way, the end result is reduced protein production, which means the gene expression is effectively downregulated. And interestingly, this binding does not need to be perfect. Even partial matching is enough for regulation, which actually gives microRNAs a lot of flexibility in controlling gene expression across different targets. Now, let's look at how microRNAs select and regulate target genes. So, the key to specificity lies in a small region called the seed sequence. This is a short but very important region of the microRNA, and it is what actually recognizes the target messenger RNA by complementary base pairing. Because of this, a single microRNA can regulate many different genes at the same time, enabling it to regulate gene expression across different pathways within the cell. And it also works the other way around. A single gene can be controlled by different microRNAs, which leads to more precise and complex regulation of gene expression in the cell. So, what you get is a very complex regulatory network, and the final result depends on the cellular context rather than on just one molecule alone. Next, let's look at how the cell controls microRNA levels. So, microRNAs are not produced constantly in the cell. Instead, their production is tightly regulated from the very first step of transcription, ensuring that they are expressed only when and where they are needed. Then, even after they are produced, each processing step can be regulated so the cell can modulate the pathway by either slowing down or speeding up these steps, depending on its needs at that time. And once they are active, their stability also plays an important role. Some microRNAs are degraded quickly, while others remain stable and persist in the cell for longer periods, which also affects how strongly they can regulate gene expression. And something really interesting is feedback loops. Sometimes microRNAs regulate genes that are also participated in controlling their own production, creating a self-regulating system within the cell. Let's bring this into biological context and explore their role in disease. MicroRNAs are contributing to normal development. They play an important role in helping cells decide their fate, guiding processes like cell differentiation and tissue formation. But when things go wrong, they are also responsible for diseases like cancer, where this regulatory balance becomes disrupted, leading to abnormal gene expression and uncontrolled cellular behavior. Because they are relatively stable and detectable, they can be used as biomarkers in blood or tissue samples, helping us identify and monitor different disease conditions. And beyond that, there is growing interest in designing therapeutic approaches that either mimic or inhibit specific microRNAs with the goal of restoring abnormal gene regulation in disease conditions. Finally, let's look at current research gaps. So, even though we know a lot, a major gap is still predicting which genes a microRNA regulates in real biological systems, since these interactions can vary with cellular context. Another issue is tissue specificity. We still do not fully understand why the same microRNA can behave differently in different cell types, showing different effects depending on the cellular context. And when it comes to therapy, delivery is still a major challenge. And finally, we really need better systems level models because microRNAs do not act alone. They function as part of large, interconnected regulatory networks within the cell. For further study, I would recommend reading the cited articles. Thank you for your attention, and I appreciate your time.